Hi, everyone. Welcome to episode 103 of Cell and Gene, the podcast. I'm your host, Erin Harris. And in this episode, I'm joined by Dr. Alexandra Snyder, Executive Vice President of R&D at Generate Biomedicines. If you've been following the evolving role of AI in biotech, you'll definitely want to listen into this conversation. Alex shares how Generate Biomedicines is using generative biology to redesign the way therapies are discovered and developed by leveraging a platform that integrates machine learning, structural biology, and high throughput experimentation to accelerate and optimize drug creation. We take a pretty deep dive into their cutting edge work on armored CAR T therapies for solid tumors and how they're addressing challenges in tumor microenvironments and why their approach allows for a broader and more efficient exploration of therapeutic hypotheses. Alex also demystifies some of the hype around AI in drug development. She offers a really clear-eyed view of what it can do and what it can't do, and how Generate is working to bridge the gap between that promise and patient impact. All right, listeners. And so with all of that background, here is my conversation with Generate Biomedicines, Dr. Alex Snyder. Enjoy. Hi, Alex. Welcome to Cell and Gene, the podcast. Thanks for your time.
It's great to be here. Nice to see you, Erin.
Thanks. So, all right, tell us about Generate Biomedicines. Talk to us about the company, its mission, and then tell us a little bit about your role as EVP of R&D.
Thanks, Erin. So Generate is a company that is trying to do what its name says. So we are really trying to reimagine how biologics are developed in order to address the greatest scientific and clinical challenges that face the field. So as you know, the field has advanced by leaps and bounds over the last 30 to 40 years. And so we're trying to identify how AI and machine learning and how the integration of wet and dry lab techniques can lead to yet another leap or bound. So our ultimate goal is to, as it says in our vocation, is to instantly generate medicines. Now, of course, what does instantly mean? How many years will it take to get there? That's a very high aspiration. So right now we're defining what are the steps to get there. How can we generate data, wet lab data, cryo-EM data, computational data, integrate those three things together to understand how to address biology more effectively and also more swiftly. In terms of how we realize that vision, we're starting with biology that's quite well known, well described, so we could really test the platform. And so we've used techniques to develop potential medicines against known targets like TSLP, IL-13, and SARS for our first molecules. Moving on from there, we're moving kind of into bolder spaces. So we have a CAR effort that perhaps we'll talk more about today. We have another effort in cancer. We're actually trying to bind to the small molecule payload that's released by antibody drug conjugates to try and protect people from the adverse effects caused by that free payload in the circulation. Moving beyond there into the future, we're looking at trying to get into cells and manipulate the biology within them, trying to target multiple things on different cell types. So we're really taking a running jump towards that leap by going through biology that's well known into areas that are less well known.
Excellent. That's so fascinating. And yes, we certainly will get into the CAR component of what you're doing. And I guess perhaps now is a great time to do that. As you're approaching the design and development of armored CAR T therapies to overcome immunosuppressive tumor microenvironment in solid tumors, talk us through how that's actually happening and talk us through that, what that is and what that looks like.
Yeah, absolutely. So I think one of the challenges, one amongst many in the CAR space, has been how do you integrate multiple hypotheses into what needs to be one molecule that you're actually testing in clinic? And so the way that we've applied AI and machine learning to the CAR space is to actually manipulate different aspects of the CAR independently and concurrently and at high throughput to try and get to what we hypothesize to be the best CAR preclinically to then move into clinic. So more specifically, often when CARs are being made, a certain binder or a certain transmembrane domain or a certain intracellular signal domain will be used, and then a armor may be added to something that's already known, and that's a fine method to use. What if, however, one could adjust each aspect of those concurrently? And so basically what we've done is to develop a suite of assays that can be performed at high throughput, looking at things like CAR proliferation, tumor cell killing, exhaustion markers on the CAR, what cytokines are released, how many CARs are expressed per cell, these sorts of parameters that we know matter to how a CAR performs in clinic, and assess them, all at once. So by doing this at high throughput, we can say, okay, let's use one binder, but change the transmembrane domain. Let's adjust the binder and fix the transmembrane domain. Let's adjust both at once. So we can actually test, those are examples, of course, you can adjust other things. By doing that and by measuring all the parameters I just mentioned together, you can really explore a much broader space of hypotheses and figure out which changes are actually making a difference to CAR function and performance and which are less important. Now, to the question about armoring that you asked, I think the key challenge there is there are limits to what we can really do in vitro. And so I think we have pushed those limits as far as we can, but we also acknowledge that the ultimate test is how these things perform in humans. And so to that end, in addition to using our own platform and our own testing, we actually do a lot of what we call reverse translation. Look at what's going on out there in the field and incorporate that into our own modeling. And so when, for example, you may have seen Carl June and his group's paper come out about the IL-18 armored CAR,
Sure.
that seemed to be effective in the post CAR failure setting. That is really informative in a way that no experiment done in a dish or a mouse could really be. So I guess what I'm getting at is we're very excited about what the platform can do to get us to what we think is the most potent, most potentially effective CAR. We also acknowledge that the ultimate test is what happens in patients.
Yeah, sure. That makes sense. And perhaps then talk us through the unique advantages associated with this. So, what is generative biology offering in engineering these armored CAR cells compared to, say, traditional design approaches?
So I think it's this ability to measure all the parameters that you want and adjust them at the same time that's unique. So as you and listeners may be familiar with, typically these variables are changed in succession rather than concurrently. And so you kind of have to choose. Do I want more expression, more CAR expression? Do I want to emphasize what cytokines are released? What this technology allows you to do is to say and instead of or, and to say altogether instead of in succession. So I want a high expression of my CARs per T cell, and I want high interferon gamma, and I want high proliferation, and I don't want high exhaustion markers. So it's the ability to do this. Now, one aspect is doing it experimentally, right? The other aspect is how to integrate the data. So, our conventional abilities to analyze and interpret such high dimensionality data are somewhat limited. And when you use AI and ML to analyze the data, you can say, okay, here's where we were right, here's where we were wrong. What should we fix to address where we were wrong? So I think it's this ability to both generate, integrate, and then act on the data that is unique.
Yeah. Okay. That makes sense. I want to talk a little bit about Generate's AI-driven platform. Talk us through how it integrates the biological data to create the novel CAR constructs, and how are these predictions validated experimentally?
Yeah. So let's kind of begin at the beginning. We can start with two different types of starting points. One starting point is where there's already a construct that's been made or studied in the field, but hasn't been optimized. The other is where you want to bind something where the field hasn't been able to bind to it before, or you want to bind to it in a way that the field hasn't been maybe a different epitope of a given antigen or a different angle of binding or something like that. In either case, you start with a starting point from either the computer or from what's been done before. And then you say to the computer, okay, here are the parameters that I'm going to measure experimentally. And here's what I think I want them to be. Like, here's what my target compound profile is. The computer then makes a bunch of suggestions, and the number of suggestions depends on how hard the problem is. So if you're starting with a known starting point, you probably don't need as many suggestions to get to a great solution.
Sure.
If you're starting completely from scratch, then you need more. So when I say more, in the latter case, you might want tens of thousands or hundreds of thousands of starting points. In the first case, you may just want hundreds up to tens of thousands. In either case, we then actually synthesize all the suggestions that the computer makes and run those through the battery of tests that I just described to you. We then take the output of those experiments and put them back into the platform, and the platform compares what the data we got are to what the target compound profile is, and assesses where it was right, where it was wrong, where it was close, and comes up with a new set of suggestions, which we then make and test again. And so we go through those, what we call cycles, sometimes just once, sometimes several times, again, depending on how hard the problem is, to get to our target compound profile. Usually we have a wide variety of choices that we then use lower throughput technologies to narrow down to our lead candidates, just as one would in conventional drug development.
Great. Okay. Thank you for that detailed explanation. And so much has been written and discussed on AI, including on cell and gene. And so I'd like to pick your brain on what you feel are the biggest, perhaps, misconceptions about AI and generative biology in drug development, specifically, of course, for cell and gene therapy, and how is Generate working to bridge the gap between all that hype and, the real world therapeutic impact?
Yeah. Thanks for that. So it's such an important question. My view on this is that we're still hypothesis testing. We may be using different sets of tools to test those hypotheses, but we still do need to define what we're asking, how we're going to get to an answer, and how we're going to define whether a hypothesis was correct or not. And so I think it's really important in this field to actually speak to what is the hypothesis you're testing, what is the AI approach lending. It is important to say what it's lending that perhaps you couldn't do with conventional approaches like you and I have just been discussing. Although honestly, these techniques are so heterogeneous and are being so diffusely incorporated now that question is almost becoming moot, because people are using AI and ML in so many different ways. But anyway, to the extent one can, I think one should define that. And so just zooming out, when we talk about what we're using AI and ML for, we think about picking the right target, creating the best drug, and then doing the best clinical experiment. Our technology is quite focused on that center piece, creating the best molecule to address the biological problem you're going to try to address. There are others who are really leaning into that first bucket, identifying the right target or targets, and that third bucket, doing the right clinical experiment and using AI to support that. And so I think it can help us negotiate some of these misconceptions by saying, okay, which part of the process are we applying AI and ML to? If the outcome is successful, great. If it's not, why? Is it something about the AI and ML? Or, for example, a company might come up with a great molecule to address a problem, but if it's not a great target, no matter how good the molecule is, you're not going to show
Sure.
success. And I think a key piece that's not discussed as much as I wish it would be, the clinical experiment is really what defines whether you see success or not. And if you think to AACR that happened recently or ASCO that's upcoming, you see this all the time, especially in the cell and gene therapy space, the T-cell engager space, how you conduct that clinical experiment. What patients do you choose? What selection markers do you use? What are you considering success? Are you looking at all the tumor sites in the case of solid tumors, or are you using just RECIST? These aspects are so important to adjudicating whether the molecule worked and whether the application of AI and ML worked.
Yeah, sure. Okay, finally, before we wrap up, I want to pick your brain on, in the 5 to 10 years, although 10, that seems like a very long time, in the maybe perhaps short term, how do you see the convergence of AI, synthetic biology, and cell therapy really transforming the immuno-oncology landscape?
Well, I love that question because, first of all, I really hope that it does. I think, I still remember when the initial checkpoint blockade data came out, how we all realized, oh my gosh, everything has changed.
Mm-hmm.
And I really hope that we're on the cusp of another one of those moments. What I'm perceiving just in the last 2 years is a shift from, are you using AI, to how are you using AI?
Right, yeah.
And so I see that convergence between AI, synthetic biology, and cell therapy happening. Where I actually feel there may be an Achilles heel is not actually in the science. It's in the waxing and waning interest in the field by funding sources and external forces. And so I think what we really need as a foundation to see that success is a regulatory landscape that is open to dialogue, that is creative, that is collaborative, and a funding environment that's willing to continue to support these efforts. For those of us who are in the space on our end, I think we need to continue to be as bold as we can in defining those 3 buckets and collaborating together, because as I mentioned, to be successful, you need to get it right in all 3. You need the right target, you need the right molecule, you need the right clinical experiment. And as you've seen, there's been a trend to test new technologies where the target is well known, which is very similar to the approach we've taken, as I described at the beginning. But now what's happened is there actually isn't as much in the new target space as I would like to see.
Mm-hmm.
And so I hope the applications of AI to that space helps kind of the rising tide lift all boats in the good molecule, good clinical experiment space. Concretely, provided we have the regulatory and funding collaboration that we need in this space, what I would like to see in 5 years is radical steps forward in the solid tumor space. Mm-hmm. That's where I think the real next breakthrough needs to come. And we've seen signals burbling, but I'd love to see a real burst in that space.
Yes, same here. And I think, there have been several subject matter experts like yourself who have echoed the sentiments on cell and gene as well. So you're certainly in good company with that take. All right, well, that's a wrap on this episode of Cell and Gene, the podcast featuring Generate Biomedicines, Dr. Alex Snyder. Alex, thank you so much. This was extremely insightful, and I really thank you for your time and all the great information you shared with us today.
My pleasure.
Thanks for tuning in. Be sure to visit us at cellandgene.com, and we will see you next time.